So Petri Saviranta from VTT Technical Research Center of Finland will introduce you to the ArrayInWell multiplex assay platform for the analysis of microbial and other antigens.
Hello everybody, I'm Petri Saviranta from VTT Technical Research Center of Finland, and I'm going to present our ArrayInWell multiplexed assay platform that we have developed in the HEDIMED and for the HEDIMED project. And together with me, there is a team of other researchers who have been involved in the project. This is a multiplexed assay platform that we have developed for high throughput multiplexed assays. The intention here is really to be able to study large cohorts which produce thousands of samples, and to study the samples for multiple biomarkers at the same time. And in HEDIMED, we concentrated on the study of the association of microbial infections with immune-mediated diseases. Well, why does it need to be high throughput and multiplexed? Because when you try to establish these disease exposome associations, it requires a study of very large number of patient samples in order to establish statistical significance. And you need to screen for multiple different factors that could potentially be important in the diseases. In this project, there are thousands of blood samples that have been collected in the different cohort studies. Also, we are interested in tens of different microbes that are of potential interest in this context. So the research questions which we want to answer with our multiplexed assay technology is: can some microbial infections, either in the mother or the child, contribute to the development of immune-mediated diseases, as have been described already in the first session? We want to link the infection history of the subjects, either the mothers or the child's, with the clinical data of the immune-mediated diseases. And in order to look at the infection history, there are, of course, several methods that are used in HEDIMED. But with our platform, we are looking at the antibodies as indicators of past infections. To do that in an efficient way, we need to have a multiplexed assay tool where we can measure many different microbial antibodies from each sample. And here is a list of microbes of interest that have been selected for our panel, so-called 40 panel, because there are a little over 40 different microbes that are interesting candidates to find these associations. We develop a workflow to study the antibodies, which starts with the cohort owners. So from different places, the cohorts, they have stored their samples in biobanks, and then they send the samples as diluted formats to VTT, where we did the analysis of the samples using our ArrayInWell technology. I'm not going into details of the ArrayInWell technology, but basically it's a microtiter plate where each well contains an array of spots that recognize different microbe-specific antibodies in the blood sample. It's based on fluorescence, and it produces images that can be analyzed with an AI algorithm using deep neural networks. We analyze the intensity data, and then we process it, and we send it back to the cohort owners, who then can use the raw data for their data analysis and interpretation. Here is a picture of our AI-based analysis tool, which can analyze the multiple spots. And this image is just from one well of a 96 well microtiter plate. So as you can see, there are multiple spots, and that can be analyzed to find out what different infections a subject has sustained in their past. For example, here it appears that among the other infections, there has been an influenza infection because we can see anti-influenza antibodies. Okay, so the results of this multiplexed assay tool, thus far we have already analyzed over 3,500 clinical cohort samples, and we have analyzed them for over 40 different microbes, and we also can look at two different types of antibodies, so IgG and IgA, which may have different roles in the immunity. So that's why it's important that we can look at both types at the same time. The association of the infections with immune-mediated diseases is being analyzed by the clinical research of the HEDIMED consortium. So I'm not reporting on the analysis. We are just we have been developing the technology platform for the use of the HEDIMED researchers. And just a few points about the policy relevance. Of course, infections are important modulators of health, but there is not enough knowledge about how different infections can lead to different chronic diseases. A better knowledge would, of course, lead to better prevention. For example, we could target certain microbes for vaccines if we knew which microbes are the most important in, for example, triggering type 1 diabetes. And the array-in-well multiplexed assay is really a tool to study very large clinical cohorts. And what's next? We are still refining the technology and our analysis pipeline. We want to have a faster throughput. We want to have really an industrial scale manufacturing of these arrays so that we can produce more of them and to have very high lot-to-lot consistency. And new cohort samples are still coming in from our collaborators, and some of them are going to be analyzed outside of the HEDIMED project, so there is already some future for this technology. There are also some calls, for example, from the Innovative Health Initiative Call 11, Topic 1. They have recognized the importance of this topic because the topic name is Understanding How Infections Foster and Induce Non-Communicable Diseases. And I think this would be a very good continuation for the use of this technology. So I want to thank our collaborators for the clinical samples and for this very interesting project. With that, I want to thank you and answer any questions.
Thank you very much, Petri. There's actually two questions. So first of all, Jenni Lehtimäki is asking, I was wondering, temporal aspect in multiplex assay of virus infections, how long antibodies stay in the blood, especially in infants who have a little different immune responses?
Well, I think there are better experts on how long the antibodies stay, but at least in this project, the samples are taken from multiple time points, and that it will be easy to identify when a new infection has occurred.
Thank you. Maybe Heikki wants to comment.
Yeah, I could maybe comment this question. So the maternal antibodies are transferred through the placenta, and their half-life depends on the levels. So higher the levels are in the mother, the longer they stay in the child. I think the practical biological effect lasts at least until the age of 6 months, sometimes until the age of 9 months. And these antibodies can protect the child against infections. So that's like the rule of thumb, I would say. And of course, sometimes the mother has no antibodies at all against certain microbes. So then the child is not protected against that virus or microbe.
Thank you. And then there's another question from Olimi Expedito. At what precision and accuracy does the AI model predict the samples?
The AI model is used to analyze the fluorescence images, which contains tens of these spots, and it's just a tool for us to be able to easier locate the spots in the images It has a very good accuracy. It's better than doing it manually, although we always do a manual final check.